Okay, great. So welcome to the first Journal Club for 2023. We're very excited to start it again this year. And our very first speaker is from UKZN at the Department of Genetics, Dr. Kwoji. And he's going to tell us about proteomics and metabolomics analysis of two co-cultures. So I'm not going to try and pronounce those co-cultures there on the slides there. You can tell us what they are. But we are very eager to hear about your research, sir. So I will hand it over to you. Thank you very much.
Thank you so much, Shane, and thank you to the organizing committee for giving me this opportunity to actually present my work. It's the journey, part of the journey of my PhD studies at the Department of Genetics, University of KwaZulu-Natal. And I will go straight into the presentation. It may not be the perfect one because, but I'm going to go ahead with it. Like it's written there, it's Proteomics and Metabolomics analysis of the cell-to-cell crosstalk between Limosilactobacillus reuteri, ZJ625, and Ligilactobacillus salivarius in ZJ614 in a co-culture. So briefly, this isolate or the strains of bacteria I used for my studies are actually members of the lactic acid bacteria, and they belong to the important families or important species, so to say, of lactobacilli. Formerly, they are known as Lactobacillus reuteri and Lactobacillus salivarius until 2020, when the nomenclature actually changed. And these isolates are important because they have potentials as probiotics. We know what probiotics are. They are beneficial microbes, which when consumed in adequate amount, confer a health benefits to their host. Some of the actions of probiotics include the stimulation of the antibodies. That is, they aid in the integrity of the intestinal barrier against pathogenic microbes. They also displace pathogens because they competitively attach to the epithelium of the gastrointestinal tract. And they also secrete bioactive compounds such as bacteriocin, which inhibit other pathogenic bacteria, and they modulate the gastrointestinal microbiome. We all now know that the gastrointestinal tract is inhabited by trillions of microbes, which with a lot of them actually being important to the host health because they aid in the breakdown of macromolecules, especially polysaccharides within the gastrointestinal tract. So this studies was actually born out of, birthed out of a sheer curiosity to understand the mechanisms of action of these two isolates, Lactobacillus reuteri and Lactobacillus salivarius, when they were administered in a co-culture. A previous study was actually carried out in my lab, and it was realized that these two strains, when they were administered to the experimental animals, they confer better health benefit compared to when they were given to these animals in a single culture. And we became curious to want to know what could be the reason. Is there any sort of interactions that is actually taking place between these two strains when you administer them to the animals, or do they kind of give an additive effect or synergistic effect to complement the action of each other? This is because we know that probiotics are host-specific, they are disease-specific, and they are also strain-specific in their actions. So we went ahead to check for the safety of these two potential probiotics. I didn't make mention of this, but then the isolate were originally isolated from South African indigenous winged snare piglets. And those piglets have history of very good health post-weaning. That is, they don't come down with post-weaning diarrhea, and they have good growth rate and better feed conversion. So these isolates were actually gotten from them. And in order to ascertain if they are real probiotics or they carry the potential to be good probiotics, they were checked for abilities to transfer virulence markers and also antimicrobial resistance. They are also checked for cell surface hydrophobicity and auto and co-aggregation, which are very important properties of probiotics, because probiotics must be able to co-aggregate or auto-aggregate among themselves in order to cause the modulation of the gut microbiome. This was carried out by Dlamini et al., who was a master student before I started my PhD. After ascertaining that these two isolates are potential probiotics, they were retested in this in an experimental animals, that is in the same piglet, to check for the growth performance of this piglet and to also check for blood parameters to check to look at the level of their stimulation of anti-inflammatory cytokines and other factors which are important immune markers, such as also antibody stimulation. So we now came down to the question that I raised earlier. Why are the co-cultures showing better effect compared to the single culture? So this gave birth to my research studies when I started my PhD, which was actually to study the cell-to-cell interactions between Limosilactobacillus reuteri and Ligilactobacillus salivarius in co-culture, but using a multi-omics approach. I only picked the proteomics and metabolomics aspect for the sake of time and not to go into, So I have a chart here to look at the aim of my studies. Like I said, is to study the cell-to-cell interactions between the two isolates. The specific objectives were to formulate defined media. Why do we need to define to formulate a defined media? As you can see under the first one, let me pick a laser. I think that would be better. Just a minute. Okay, yeah. So we went ahead. Okay, it was important for me to actually formulate a defined media, because in order to do analytical studies, we don't want to have confounding factors. This is because the enriched medium, which are normally used for the growth of bacteria, contains undefined compounds. For instance, the source of proteins in an enriched medium could be beef extract, and then or peptone extract. And the source of vitamins could be yeast extract. So doing metabolomics or proteomics studies with such kind of compound that you don't know what you actually are feeding the isolate could result in some proteins or in some metabolite which were not actually from the isolate, but could be from the medium. Hence the rationale for formulating a defined medium. And after formulating the defined medium, I went ahead to also determine the nutritional need of this isolate by carrying out what we call single omission experiments to come up with a minimal defined media. Because remember, in an ideal settings, this isolate might not be exposed to what they really need for their own metabolism or for their optimum growth. So we want to reduce as much as possible the amount of nutrient that we're giving them. That was why I now formulated minimal defined medium, and I did a series of experiments. I'm going to show the data on that one when I come to it. After formulating the minimal defined medium, I also went ahead to do a growth kinetics to kind of get an idea of the growth pattern of these two isolates when they are in single and also in co-culture, so as to know at which point I'm going to take my samples for both the transcriptomics, proteomics, and then the metabolomics analysis. The second objective was to do metabolomics, of course, and I prepared the samples for untargeted metabolomics. That was what was done. And the rationale for doing that was to determine the functional metabolite, also to determine differential metabolite and do functional analysis and pathway enrichment in order to see the variation of the metabolite that are being expressed by these two isolates when they are in single culture and also when they are in co-culture, and to be able to have an idea of what is actually happening at the co-culture that makes them different in terms of their behaviors when they were administered to these animals in single culture. The third objective was to do proteomics. So for the proteomics, I actually did intra and extracellular proteome, that is both the secretome and then the intracellularly expressed proteins. Of course, the rationale too was to determine the differentially expressed proteins, to do post-translational modifications, to see if there is any of these post-translational modifications, because we know that post-translational modifications affect the functions of proteins and enzymes, to do pathway and network enrichment analysis and do protein-protein interaction to determine the level of interactions between these proteins. And the last objective was to do transcriptomics, but I'm not going to talk about the transcriptomics yet in this presentation. But then, yeah, like I said, the whole project was a multi-omics approach to understand the nature of the cell-to-cell interactions between these two isolates when they're in a co-culture. So for the chemically defined medium, here is the result. And like I said earlier, I did what we call leave one experiment or single omission experiment. And in single omission experiment, what happened is that I had a total of about 48 compounds, which consist of the 20 amino acids, 10 vitamins, yes, I think 10 or thereabout. And I also have the 5 nucleoside, 5 nucleoside bases, and also other minerals such as the phosphate buffer. Okay, I have the table having all of them. So briefly on the leave one experiment, after culturing the isolate on a medium which is devoid of amino acid, we can see this AA indicates amino acid, which denotes that these two isolates are actually auxotrophic for amino acid, which means that they cannot go on medium without amino acid. Hence, I went ahead to check which of the amino acid is essential for the growth of the isolate and which ones are not essential. As you can see on these figures, this is figure B for the leave one experiment for the amino acid, and then figure C is the leave one experiment for the vitamins, and D for the nucleoside bases. So in more detailed form, the defined media formulated for L. reuteri and L. salivarius show that the isolates, particularly if we look at from the first column now from the left is the CD, and that is the complete defined medium containing all the nutrients. The next one is the minimal defined medium that supported the growth of Lactobacillus salivarius, and we can see some of the minerals are actually not essential for the growth of the isolate, hence they were omitted in the minimal defined medium for Lactobacillus salivarius. The same thing, if we go down, we will see some amino acid like alanine, proline, lysine, and lysine, and also nucleic acid bases like guanine and adenine, not essential for the growth of this isolate. The same thing for Lactobacillus reuteri. But however, since I am culturing the two isolates together in order to determine their behaviors in a co-culture, I had to come up with a consensus medium that will support the growth of the two isolates. Hence, you will see that some of the compounds which are not essential for one being appearing in the minimal defined medium for the two isolates, so as not to deprive the other from growing. Like for instance, L-alanine is not essential for Lactobacillus salivarius, but then it was added in the consensus medium. The same thing if we go down to, I think that is all for the amino acid. But then for the vitamins, you will see, for the nucleic acid bases, you will see that adenine is not essential for reuteri, but then it was added in the consensus medium in order not to deprive one. And the same thing in the vitamins, I have some as well. So this is a growth kinetics of the two isolate. Like I said, I generated growth curve data using an automatic spectrophotometer that took reading at interval of 15 minutes. And like I said earlier, the essence of this is for me to determine my sampling size and not to go into the details of the mathematics that actually took place here. But then we realized that the logistic LAC2 model, I'm not too much into modeling, but yeah, as part of this manuscript, and I had to do it on Python. Yeah, so because of the and the model with the lowest BIC was the one which is the perfect fit for the growth of the isolate. Of course, like I said, I don't have much to do with the modeling, but I only this to determine my sampling point, at which point I'm going to do my sampling. We'll also realize from this data that, okay, I have a consensus one with both the two isolates, so I will quickly go there so that I will do the comparison. But unlike for the L reuteri for L salivarius, the Baranyi-Robert model was the one which was the perfect fit. And like what I was trying to explain earlier, we can see that from the growth curve, L salivarius have a prolonged lag phase compared to L reuteri. And then we also realized that L reuteri have higher growth rate, that is generation time is higher compared to L salivarius, which means that L reuteri have the potential to outgrow L salivarius when they are in a co-culture. And of course, even some of the phenotypic properties of L reuteri showed me that, because it's one isolate that produces a lot of reuteri gas, and I saw that too on the course of my studies, although it was outside my scope to look at the properties of that gas. So I really didn't pay much attention to it, even though I wanted to. Yeah, so this data had been published, I think, since 2022 earlier, yeah. So I will move to the metabolomic analysis. For the metabolomic analysis, like I said earlier, I cultured the isolate in single and then in a co-culture. And I also did a very bulky work in the sense that I did the metabolomics and the proteomics and transcriptomics analysis for both the isolate on the chemically defined medium, the complete defined medium, and also on the minimal defined medium. As you can see on the VIP scores here, I have CLR, CLS, CLR, LS, indicating the isolate on the complete chemically defined medium, whereas MLR, MLS, and MLS-LR indicating the isolate on the minimal defined medium. So this is showing the differential metabolites which are expressed by this isolate. Like we have pyrimidine as one with our then 4-aminobenzoic acid, arabifuranose, phosphoric acid, D-mannose. These are some of the metabolites which are differentially expressed between the groups and also within the groups. This is just a heat map also showing the differential metabolite. Like I said, we have N-acetyl glutamic acid, oxopentanoic acid, butanoic acid. These ones that I didn't indicate what they are actually those which are not yet annotated, if that is the correct word to use. Yes, they were not actually annotated, and we don't have identities for them so far. But then these ones that I've highlighted are the ones which have been identified, and I did the functional analysis as well. So this is just a PLSDS score showing the clustering of this isolate both in the complete chemically defined medium, as we can see on my left, and also the complete, the minimal defined medium. You can see that the isolate actually clustered based on the medium types. This is a classification, metabolite classification and pathway enrichment analysis showing that the major groups of the metabolite excrete are produced by this isolate falls into the monosaccharide, amino acid and peptide, pyrimidines, benzoic acid, and majorly they are associated with lactose synthesis, glycolysis, fructose and mannose degradation, gluconeogenesis, and these are some of the important metabolic pathways that the metabolites are associated with. I'm going to revisit this pathway this pathway map when I discuss the proteomics, because I would like to discuss it once at a time. So this is for the proteomics analysis, and like I said, for all the analytical analysis I did for both the complete chemically defined medium and then the minimal defined medium. In the case of the proteomics analysis, like I said earlier, I did both secretomics again and intracellularly expressed proteome. So the figure A now is showing my internally expressed proteome, and we can see the summary of the total proteins which are expressed between and within groups for internally expressed proteins on figure A, and extracellularly expressed protein, that is the proteome, the proteins which were isolated or which were identified in the medium that were secreted by this isolate. That is what I mean by the secretomes. So importantly, functions of some of the differentially expressed proteins is that we were able to identify some important proteins, especially that varies when the isolates were co-cultured and also when the isolates were singly cultured. Like S-ribosyl homocysteine, which is log X, was higher in CLR-LS, that is in the co-culture, compared to in the single cultures. This forms a vital aspect of my thesis, because with this I was able to see one of the mechanisms by which these two isolates interact or crosstalk when they're in a co-culture, and that is through what we call quorum sensing by the secretion of autoinducer 2. This quorum sensing is actually a mechanism by which bacteria interact among themselves in order to regulate cell density and also inform each other about the metabolic potential of the environment. Quorum sensing is also one of the important mechanisms by which bacteria cells interact with the host epithelium when they are within the gut. So this is very, very important, and it shows that there is actually interaction taking place when the two isolates were co-cultured compared to, and of course it is expected, because if it is single culture, why would the isolate want to regulate its own population? Why would it want to, what signal would it want to send to who? So this is a form of synergy, which means that these isolates tend to regulate their growth and also send these signals to communicate with other important microbes, because we know that probiotics, one of the functions of probiotics is to regulate or to modulate the gut microbiome, to aid the proliferation of beneficial commensals and suppress the overproliferation of pathogens. Hence, when there is an imbalance, what we describe, what we know as dysbiosis, probiotics are actually being administered in such cases in order to restore the gut which has been destabilized either due to intake of antibiotics or other factors. So another important enzyme is phosphoglycerate kinase, which is important in carbohydrate degradation. That was why I said I was going to leave that pathway to come back to it, because it is interwoven. And until when I did this, analyze the proteomics data, I never appreciated the metabolomics data alone. Another important protein is enolase. Of course, these are not just this is not, I have tons of, this table is a very, very large one, but just for the sake of presentation, I normally just highlight on this very few ones, because we all know how large metabolomics and proteomics data are. So the protein-protein interaction network actually shows that there is a very high level of interactions between the proteins, in fact, beyond what is expected, meaning that these proteins belong to virtually the same family, like in case of those that are associated with the carbohydrate metabolism, and those associated with lipid metabolism, and also some which are associated with nucleic acid synthesis. So these are just further data showing the protein-protein interactions enrichment. And like I said earlier, we have translation factors, also very visible elongation factors. These are some of the differential proteins which are secreted between the two between the co-culture and then the single cultures, and majorly for the single culture and also for the proteome, internally excreted proteins, and the secretomes, the proteins falls within the cellular anatomical entities, intracellular proteome, and then cytoplasm. The same thing applies to all of them. They fall within the same group, meaning that this is actually not from just the medium, but then it's something which was coming from inside out. Yeah, so this is by showing the biological functions of the proteome and then the secretomes. That is for the differential proteomes and the differential secretomes. So metabolic process, these are just the biological functions of the identified proteins. Some of the post-translational modifications identified are transcription anti-termination, that is a deamidation, which was also found. We also identified oxidation and dehydration, that is loss of water molecules, and then the removal of ammonia. So like I said, I was going to come back to this chart before I finally conclude my presentation. Both the metabolomics and proteomics actually show, like the proteomics confirm the excretion or the secretion of phosphoglycerate kinase, which are enzymes associated with the breakdown of glucose, which was also high in the co-cultures, and alpha-D-glucose 6-phosphate is a metabolite which was also identified. So there is actually a kind of agreement between these two data, and showing that the majority of the metabolite and then the proteins are those which are associated with pentose phosphate pathway, glycolysis, gluconeogenesis, and pyruvate metabolisms. In addition to some of the mechanisms like I identified, that is the mechanism of cell-to-cell communication, which is through the synthesis of autoinducer 2, important in quorum sensing. So in conclusion, the defined media that I formulated actually supported the growth of the strain, and the co-culturing of reuteri and salivarius influenced their proteome and metabolite profiles, which indicates that there is a level of interactions between them, between the two isolates, and important enzymes such as phosphoglycerate kinase and enolase that plays roles in the breakdown of glucose were highly expressed in the co-culture, and homocysteine layers, like I said, which synthesizes autoinducer 2, involving quorum sensing, was also identified. There are also other compounds such as the elongation factors, glyceride 3-phosphate, which are important in lactobacilli attachment to the host epithelium. However, I am also being very careful with these two last conclusions because my studies was purely an in vitro studies on a synthetic defined medium. So hence, I try to be careful when I make this conclusion, because the situation might not really be the same were it to be carried out on cell or tissue culture, which are some of my recommendations, and if possible, an experimental animal studies should be carried out. So this is in conclusion to part of the stories of my PhD journey. I don't know. Yeah, this is the end of my presentation. Thank you for listening.
Thank you so much, sir. That was an awesome presentation and a great benchmark to start our journal club at. So I'll open up the floor to questions in the group. So as last year, if you have any questions, you can just raise your hand and you can ask directly verbally to our speaker, or you can write in the chat box. I would also like to ask you, Dr. Kwoji, to just share your email in the chat box.
Okay.
So I'm sure anyone who wants to get in contact with you after the talk to discuss your project, maybe collaboration, or someone who wants to do something similar. It's always good to get communications open between speakers and the audience. But it's great to have a talk on probiotics, because I haven't had one yet. And yeah, functional health is a big thing at the moment. So this is an important area of research. And you did quite a lot of work for your PhD. proteomics, metabolomics, and a very meticulous approach to your media as well is very impressive.
Thank you.
I know I have a student of my own in the audience that was doing proteomics, metabolomics, and it'd be good for her to see how we can use proteomics and metabolomics together. It's very interesting.
Yes.
So are there any questions from our audience for our speaker? There is a hand from Mr. Mshlongo. You can unmute.
Okay, I'm not very good with names, but greeting colleagues and friends. Kwoji, is this an LC or GCMS data? I think I missed that component. And my second question is a bit of a long question involved in the crosstalk of microbes and everything like that. You mentioned that you had controls which were the species grown alone, and then you have them combined. And I'm just interested to see if you find any biomarkers on the controls that you could use that could specify the metabolites perturbation related to the species differences. So by that, I want to know if you had any biomarker that you can say when it's produced by the species alone, is it still produced when you have them combined? And how is it affected by the combination? Number 3 is the current sensing where you identified the enzyme. I've forgotten the enzyme, but when I look at your data, there was no compounds that are related to that current sensing. Do you know why you couldn't identify them, or they were part of the metabolites that you couldn't identify? Hence, I'm asking if this is LC or GCMS data.
Thank you. For the metabolomics, it was GCMS data. And for proteomics, it was LCMS data. Then coming to the questions on the biomarkers, actually, there was no any biomarker which I used in order to show, but it would have actually been very nice to have that, because that could have opened up my data for clarity, sort of. You know, I wouldn't have to be struggling with trying to identify the levels based on the levels now. So we actually didn't have any biomarkers to say that, okay, with these biomarkers, we are going to make this kind of conclusion to see if this is produced in the single culture or whether it is not or not produced. So for my data, what I was able to see was to check the level of to check the level of what I said now, production or expression now of the respective metabolite proteins. Then for the you asked question on the quorum sensing data, the quorum sensing one, the enzyme which I identified, it was revealed from the proteomics. One thing I wanted to do again was to kind of do a 2-level or 3-level kind of analysis to be able to follow the data from proteomics to metabolomics, to have a kind of network to see if I will be able to have a flow, to understand the flow of information between these 2 different levels or 3 different levels, like I do have now. But I have not yet done that. And of course, like I have so much time not on my side, I've spent a lot of time doing this work, and I had to submit my thesis, but I'm still doing like the transcriptomics data. I'm still working on the transcriptomics data, trying to analyze it and process the manuscript and stuff like that. Yes, I don't know if I've answered your question, but yeah.
Okay, no, thank you very much. You said that you used GCMS, ne?
Yes.
And I might be not fair, but I want to know, was this derivatized or it was just extract with a solvent and then ran on the GCMS? And the libraries that were used to assist with the annotation, do you have information on that?
I do have information, but I think limited information because I outsourced it. I didn't do it myself. So they were derivatized, I'm sure they were derivatized because I sent it to Northwest, and yeah, Dr. Ilse Dupriz has done an in-depth, they were derivatized. That is one thing I know. From the data, I know that they were derivatized. And what next did you ask? They were derivatized, that I know.
Okay. The other question was the libraries used for the identification, but since you outsource,
Yes,
you don't have that information.
Yeah, she did also the identification, the metabolite identification, So I only did the downstream statistical analysis.
Okay, no problem. Thank you very much.
Thank you.
I think I can answer on behalf of Ilse Dupree. So I'm next to the Center for Human Metabolomics. They use the NIST libraries, same as the ones used by Otto Fifine's lab. So NIST, the NIST libraries.
Okay.
There is a question in the chat room. So one of the audience members, Loretta, really liked your infograms, nice figures, and would really like to know, where did you use the tools to generate these figures?
Okay, we have the subscription for BioRender. So I did these figures from BioRender. Yeah.
Okay.
Yes, I did these figures for BioRender. From BioRender.
So
I did them myself. Yeah, I did them myself. So
it's a very impressive presentation.
Yeah, thank you. Thank you.
Are there any other questions for our speaker? Please, we encourage questions.
I have a comment and a question, I guess. Comment is, I'm very impressed that the field of probiotics has moved to a point where people can comfortably show mechanisms of action. Back when I did my master's in probiotics many moons ago, the questions used to be more about the relevance of the work and whether we think this approach works. Was more defending the concept of probiotics than the actual experiments done. So I'm glad that now people can focus on the experiments that work, because the field is sort of proven now and it's common practice now for people to take probiotics. My question is, would you, from your preliminary analysis of your transcriptomics data, sort of give a sneak preview of what you're seeing so far, or what contribution to the study that data is showing?
Okay, for the transcriptomics data, what happened, in fact, that I couldn't add the transcriptomics data to this analysis was because, like I said, my omics analysis, I outsourced them. So I had a delay in getting the transcriptomics data. I think there was a very long queue at the MRC. I outsourced it to MRC, and time had actually gone. So far, the only information I have, I don't know if I will be able to quickly locate the folder on my desktop, but the information I only did the QC, and I was not really able to, I couldn't find time because I just concluded I had my PhD conferred just like 2 weeks ago. And I got the data sometimes in December, the holidays, and a lot of things. I really didn't go too far with the analysis. I'm at the point of doing the mapping of the transcriptomic data trying to do the genome mapping and to continue with the analysis. But for now, I really only have the QC information. That was the only thing I did. So I'm just trying to generate the library and then do the mapping and then proceed from there. So for now, I don't have much information. All I know that from the QC, the transcriptomics data, that is the analysis was okay as the, based on the QC, fine.
Okay, thank you for that. I guess you'll have to give another talk soon.
Yeah, it's okay.
Yes, the follow-up talk will definitely be needed in the future. What is next for Dr. Kwoji?
What next for Dr. Kwoji is, I am so happy. This morning, I just got my visa approved. I'm moving to Czech Republic for a postdoc. I actually been employed as a postdoc at the Faculty of Medicine to work on the influence of gut microbiome on type 1 diabetes in children. So I will be moving to Charles University in Czech Republic, which happens to be their premier university, the second Faculty of Medicine, Department of Pediatrics and Medical Microbiology. Yeah, to continue the microbiome and diabetes story there. So
that is amazing.
Yeah, thank you so much. Yeah.
Congratulations, that is amazing.
Thank you so much. Thank you.
I hope you come back to our country and share the information you learn there at the Czech.
Yeah, I will keep, I think I've always been following MSA. So I will continue to be part of this journal club, irrespective of where I am. And I don't know, but I think one of the major challenges I wanted to, because it was also mentioned here, and I actually saw it, was how publishing metabolomics data. I think there was a time someone talked about that. But then, yeah, I don't know if someone can share something about that, because I think these are some of the challenges I'm facing here. But yeah, I think we're going to overcome that. But so far from this thesis, from this work, I have already 3 publications down. One was just accepted on Monday with a Nature peer journal, the Nature Partner Journal. These are Food of Science, Science of Food. So yeah, the metabolomics data is under review along with the proteomics data. Because at first I was being asked to provide any proteomics information to support the metabolomics data. Yeah, so I think combining these 2 tools together will actually help in the future, because, yeah, thank you.
So Malati, we need to definitely contact Dr. Kwoji in the future to have him come back.
Definitely, definitely.
Thank you.
Okay. Any more questions from the audience? If there are any other questions, or you think after the talk, after we finish the presentation, or after the Zoom meeting is finished, I'm sure Dr. Kwoji will welcome any emails to discuss his project, because he sounds very enthusiastic about gut microbiome and probiotics. I'd see Mazizi has another question.
Yes, last question from me is that is some of this data, especially the selection of the media where you use the models and everything to see if the bacteria will outgrow each other and everything, and you came up with a media that was suitable for both bacteria. Do you have that published, or it's still not yet?
No, it was published since 2021. No, early 2022. Yeah, it was published. I removed I can give the citation. Yeah, but if you just Google it, Kwoji, it will definitely pop up. So yeah, it was published. Yeah.
Okay, no problem. Thank you.
Some of the this was also this was my review. As you can see, this is also my figure. It was my first review for my thesis. It was published in 2021. This particular figure here. Then this one was published. Also, this is the one for the medium. And okay, the multi-omics data also was the one which was the review for multi-omics, which was the one accepted for publication just last week by NPJ, Science of Food. And this, the metabolomics and proteomics one under review now. Yeah.
Okay, that is awesome. 3 publications and Czech Republic postdoc. We wish you all the best there, Dr. Kwoji, and thank you so much for
Thank
joining
you.
us and sharing with us your project. It was very interesting.
Thank you for having me. Thank you so much. Yeah.
And I thank everyone else for attending.